Welcome everyone. Today's session is a knowledge briefing on ICH Q5E, the guideline that explains how regulators expect manufacturers to demonstrate comparability when a biological product undergoes manufacturing changes. This deck is based on two foundational ICH documents, the Q5E Guideline, finalized at Step 4 in 2004, and the earlier concept paper from 2002 that explains why Q5E was created in the first place. The big idea you should keep in mind throughout the session is simple. Biologics are complex, and even small process changes can shift quality attributes, so we need a structured, science-based way to prove the product before and after a change remains clinically the same. Let me set expectations for what we'll accomplish. By the end of this session, you should be able to 1. Define what Q5E means by comparability and why it matters for biologics. 2. Explain the regulatory problem Q5E was designed to solve. 3. Identify the types of manufacturing changes that trigger comparability work. 4. Describe the stepwise, risk-based comparability approach Q5E recommends. 5. Understand when analytical data is enough and when you need non-clinical or clinical bridging. Think of today as a practical roadmap you can apply whenever a process change is proposed. First, what exactly is comparability? In Q5E terms, comparability is the conclusion that a biological product produced before and after a manufacturing change is highly similar in quality attributes, and that any differences detected are not expected to impact safety or efficacy. Notice what comparability is not. It does not mean the products must be identical. Biologics naturally show batch variability. The goal is to prove that the product remains within a scientifically justified similarity space tied to clinical performance. So as you listen today, keep separating two ideas: difference detected analytically versus difference meaningful clinically. Q5E is basically a framework to bridge that gap. Now the why. The 2002 concept paper makes it clear that Q5E was born out of real regulatory friction. Before Q5E, different regions, the US, EU, and Japan, had uneven expectations for change management. A manufacturer might need one comparability package for one regulator and a different one elsewhere. That led to slower approvals, duplicated studies, inefficient inventory use, and delays in rolling out process improvements. The concept paper proposed a harmonized scientific approach. Start with quality comparability, and only move to safety or efficacy studies when quality alone can't answer the question. So Q5E is essentially the international solution to a global inconsistency problem. Let's clarify scope. Q5E primarily applies to well-characterized biotech and biological products, especially proteins and polypeptides, their derivatives, and products made through recombinant or non-recombinant cell culture. It covers manufacturing changes during development and after approval. A key assumption in Q5E is that the same manufacturer can directly compare pre-change and post-change material. If you're dealing with a product outside the scope or a situation where side-by-side comparison isn't feasible, Q5E may still be useful conceptually, but you'd need to align early with regulators on expectations. Q5E doesn't list every possible change, but it groups changes into those that could affect quality attributes. Examples include changes to cell line or upstream fermentation parameters, changes in purification steps or chromatography media, facility or site transfers, scale-up or scale-down, and new equipment or new raw material suppliers, a drug product formulation, container closure, or fill-finish changes. The more upstream and fundamental the change, the higher the likelihood it affects critical quality attributes, and therefore the more extensive the comparability package. This slide is the heart of Q5E. The guideline is explicit. Comparability is risk-based. The extent of data depends on the potential impact of the change. You begin with a deep understanding of the product, the process, and its critical quality attributes or CQAs. Then Q5E recommends a stepwise ladder. 1. Analytical comparability first. 2. Then biological or functional confirmation. 3. And only if uncertainty remains, non-clinical or clinical bridging. Crucially, you should leverage prior knowledge, for example, known links between glycosylation or aggregation and clinical outcomes. So the guideline is not about testing everything. It's about testing what matters most, justified by science. Planning is where success is determined. Q5E says you should decide where in the process to compare material. You might compare intermediates, a drug substance, a drug product, or multiple levels depending on where the change occurs. Then you define the extent of comparability testing based on the nature and location of change, expected effect on CQAs, a sensitivity of analytical methods, and existing clinical knowledge of attribute relevance. Another Q5E emphasis: define similarity criteria before testing, and use historical batch trends to understand normal variability. So comparability should never feel reactive. It's a planned, pre-specified scientific exercise. Q5E lists the major quality domains to compare. You look at physicochemical properties, including primary sequence and variants, a higher order structure because folding changes can matter clinically, post-translational modifications like glycosylation or oxidation, a biological activity or potency, which ties most directly to function, a purity and impurities, including aggregates and process-related contaminants. The principle is that your analytical program must be broad enough to detect meaningful shifts, not just confirm that routine release tests pass. This slide gives representative tools. The guideline doesn't mandate methods, but it expects orthogonal, sensitive assays. For identity and primary structure, you might use peptide mapping or mass spectrometry. To detect charge or size variance, methods like ion exchange, capillary electrophoresis, SEC, or SDS page are typical. Glycan profiling often uses HPLC or MS, sometimes lectin-based assays. Higher order structure can be probed with circular dichroism, FTIR, DSC, or NMR. Potency uses binding or cell-based functional assays. And impurities cover host cell proteins, residual DNA, aggregates, leachables, etc. The key Q5E message: use multiple methods to triangulate the same attribute, because no single assay is perfect. Q5E draws a bright line here. Routine release specifications do not by themselves prove comparability. Passing specs only shows the product meets minimum quality thresholds. Comparability requires a wider characterization lens plus trend analysis against historical variability. Sometimes specs may need revision after a change, but widening specs is discouraged unless you can justify why the broader range still protects safety and efficacy. So think of release specs as a subset of the comparability evidence, not the totality. Stability is another area where subtle differences appear. Even changes that leave CQAs apparently unchanged at release can alter degradation pathways over time. Q5E expects stability comparability to use real-time stability data and accelerated or stress studies when appropriate. These are interpreted under ICH Q5C and Q1A principles. So stability isn't an afterthought. It's part of ensuring the post-change product remains comparable through shelf life. Analytical data needs to sit within a process story. Q5E expects you to show the modified process remains in a state of control. That means a revalidation of affected steps, demonstration that downstream controls still work, and comparison of key intermediates if they help interpret changes. For marketed products, comparability should use commercial-scale post-change batches, not just lab runs. And regulators want a side-by-side narrative. What changed, why, and how control strategy ensures consistency. So comparability is as much about process capability as it is about test results. Now this slide answers the question everyone asks. Do we need clinical bridging? Q5E says, move beyond analytics only when necessary. You need non-clinical or clinical studies if equality differences are detected and their clinical relevance is unclear, or analytical methods can't confidently rule out meaningful structural/functional change. The extent depends on risk factors like a magnitude/significance of observed differences, product complexity, a strength of structure-activity relationship, immunogenicity concerns, indication, population, dosing, and therapeutic window, and prior clinical experience with the product. Possible bridging includes PK/PD comparability, immunogenicity studies, or full efficacy/safety trials in rare high-risk cases. From a submission standpoint, Q5E expects a clear comparability package that includes a description of the change and rationale, a risk assessment linking change to potential CQA impact, analytical comparability data with predefined criteria, process validation or revalidation evidence, a stability comparability plan/data, and if required, non-clinical/clinical bridging results. The tone should be scientific argument plus data, not a data dump. Regulators want to see your reasoning pathway. Let's translate Q5E into day-to-day best practice. Maintain a robust control strategy and an up-to-date CQA list. Use multiple orthogonal assays for high confidence. Trend historical data to define normal variability, not just spec limits. Document risk assessment early before you test. Engage regulators proactively for major changes rather than surprising them at submission. These steps reduce both scientific risk and regulatory friction. To wrap up, 1. Comparability means high similarity with no expected clinical impact, not perfect identity. 2. Q5E exists to harmonize global expectations for post-change biologics. 3. The exercise is risk-based and stepwise. Analytics first, bridging only if needed. 4. A strong analytical battery and historical trend context are central. 5. Process evidence and stability complete the comparability story. If you follow the Q5E mindset, science first, risk-based, justified, you'll be aligned with regulators across ICH regions. Thank you for your time and attention. If you have questions, or if you'd like to discuss how Q5E applies to a specific change scenario you're working on, I'm happy to go deeper. Comment me with your queries and I'll be back with the explanation.